David Serre. David is a professor at the University of Maryland School of Medicine and he's also a member of the Institute for Genomic Sciences at their university. So his primary research focuses on malaria-causing parasites, primarily Plasmodium vivax. And his group has really been trailblazers in really using these new cutting-edge next-generation sequencing technologies to better understand the biology of these parasites. So prior to arriving at Maryland, he did his PhD in Germany at the Max Planck Institute for Evolutionary Anthropology. Did a stint at McGill University in Canada, and then spent some time at Case Western and the Cleveland Clinic as an assistant professor there prior to joining the University of Maryland. So today, I'll let David present some of the interesting work that he's been doing, and I want to thank him for joining us here to present some of his findings.
Thank you. I need to moderate this introduction. So I trained in human genetics and genomics, and then I made the switch to Plasmodium maybe 15 years ago. And the rationale behind it, the true rationale was because the genome was so much smaller, and as an assistant professor, you could actually sequence genome and afford it. But then I got really excited by the biology because these are organisms that have some really weird biology. And we can learn a lot from genomic approaches. I'm going to show you some example. Now, on the trailblazing thing, that's where the moderation come in, because I always have the impression that in parasites, the genomic approaches we use are 10 years beyond or behind what is being done on model organism and human. So don't expect something super exciting and revolutionary because you're not going to see it here. We're just trying to tweak the methods that have been developed for human and mouse and apply it to our system. I'm just going to give you a few vignettes of what we're doing. But feel free to interrupt me anytime with question, and I'm happy to talk about something else than what is on the slide if you want. So just, I'm only going to talk about malaria today. We're trying to diversify because I get distracted with some other organisms that have also some interesting biology, but today I'm going to only talk about malaria. So it's a disease that we've made a lot of progress against it in the last few decades, but it's still a major public health burden. So most people get sick, get uncomplicated malaria, which is usually chills, fever, and anemia. And in some cases, and we don't really understand why, some people get some severe complication that can actually kill the children that are affected by that. And it's a worldwide disease, so again, we decrease the numbers, but there's still like 250 million cases of malaria every year, and almost like more than 500,000 deaths, mostly in children under the age of 5 in sub-Saharan Africa. And these numbers will go up. Like we know with the current climate and the current politics that these numbers are going to increase in the next 1 and 2 years. So it is a big challenge, and it's still a big challenge, and it's going to remain like that. Half of the world's population is at risk of getting infected with malaria. And beyond the health problem, there's a major issue for society and for the economy. So in places when you have endemic malaria, you have a large fraction of the workforce that even if they don't die, are sick, are out of work, and that perpetuate a cycle of poverty. So malaria is one of the diseases that we could eradicate today if we had political and logistical will to do so. We have very good drug for it. The reason why we have malaria, political instability, is economical choices. So the fact that people are sick from malaria, don't work, creates some poverty. That poverty creates more malaria. So that's a cycle that we really have to tackle. In terms of the biology, which is what I'm interested in, malaria is transmitted by mosquitoes that are infected. When they bite you, they inject the parasite. And these are unicellular eukaryotes. Okay? And that's one of the major complications of eradicating malaria, is because the eukaryote, they shared a lot of the molecular mechanism with us. So a drug that targets well the parasite will often have some side effect on the host and the patient directly. And that's one of the major complications. So that everybody's on the same page, when you get bitten by a mosquito that is infected, the parasite gets into the bloodstream, through the dermis, into the bloodstream, migrates to the liver. In the liver, the parasite will multiply asexually, So from a handful of parasites, you get to 10,000 or hundreds of thousands of parasites that get released from the liver and infect red blood cell. And in the red blood cell, one parasite will go into one red blood cell, multiply asexually, release something between 16 and 32 parasites that will then infect new red blood cell and continue. And that cycle is what most people study. That's what's causing all the symptoms of malaria, the fever, the anemia, the chills, everything is caused by the blood stage of the infection. At some point, some of the parasites will differentiate and create these gametocytes, which are fully differentiated parasites that can get picked up by a mosquito. In a mosquito, you have sexual reproduction happening. So the parasite was haploid in the human host. In the parasite, you have meiosis, and the parasite is shortly diploid. And then the haploid parasite moves to the salivary gland and be ready to infect another patient. And so in terms of biology, we have this really, really cool system when the disease is caused by the interaction between the parasites that has many different forms, the human host, and the mosquito. So they have this very complex biology that we're trying to address. On top of that, there's not one single malaria parasite. So in humans, there's at least 6 different species that can cause human malaria. Most of what my lab used to work on is Plasmodium vivax. Now we're extending into Plasmodium falciparum, which is the parasite that causes most death in Africa. But there's other species, and these parasites are very, very different. So we don't exactly know when they diverged, but it's probably more than 60 million years ago. So they are high divergent from each other as a human is from a cow. So they both cause human malaria. They can cause the similar symptoms. Their life cycle is similar, but they have extremely different parasites. And yeah.
Could you please speak on the OK so we could read the captions better?
Try it. That we don't need. Is that better? That we not?
Thank you.
Perfect. Sorry about that. All right. What I work on is the genome, not the parasite itself. Give me some parasite. I actually cannot tell you what they are. They all look the same. I can actually not differentiate the parasite from the human white blood cell. But I know the genome. So the genome, just so that you know what we're looking at, that's one form of parasite. It's got 2 organelle genome, a mitochondria. And one thing that is really weird if you come from human genomics is there's a single mitochondria per parasite. Hurt my brain when I learned that. The genome is very degenerate, so it's only 5 kilobases long. So it's missing a lot of what you usually see in eukaryotic mitochondria. And it's linear. And on top of that, you have a second organelle, the apicoplast, that gives the name to the whole parasite family. It has also its own genome, and we don't fully understand what it does. In terms of the nuclear genome, it's a small genome, so that's easy to sequence. It's only 25 megabases, split across 14 chromosomes. And just to give you an example of how these parasites are different. That's Plasmodium falciparum, the one that kills children in Africa. It's 20% GC. It's a nightmare to sequence. Only A and Ts. Especially if you're outside genes, you're sequencing A and Ts. Vivax is the other major human parasite, 45% GC, like human and mouse. So completely different in terms of gene composition. And again, they can be haploid or diploid. One thing that complicates some of the things I'm going to talk about is in these 25 megabases, you have 5,000 protein coding genes. That makes that the genome is really gene rich. You have genes everywhere in the genome. Roughly half of the genome is coding. Okay. And the thing that really bothers me a lot, I'm trying to understand, is out of these 5,000 genes, which is a quite large number, you only have 30 non-transcription factors. You have a very limited set of transcription factors that you need to regulate to this very complex life cycle. When a parasite sometimes has to deal with the patient immunity, sometimes with the mosquito immunity, sometimes it's intracellular, sometimes extracellular, sometimes it has to multiply asexual really rapidly, and sometimes it's quiescent for several weeks. And all that regulation is orchestrated with a very small number of master gene expression regulated. We don't understand what's happening. So that's one question that my lab is getting more and more interested in. How do you regulate gene expression in Plasmodium genome? Just to give you a comparison, baker's yeast, so I hope nobody works in baker's yeast because it's got the same number of genes, but it does something that is really boring. It does nothing. Binding, and sexual reproduction. So a much more simple life cycle, but he uses the same number of genes. So what I'm really curious is how do we reconcile the phenotypic diversity that we see in Plasmodium, the different environment he has to cope with that very simple genome structure? And the answer is it's probably not that simple. So that's why we're doing that. So my lab works on two different approaches that really feed each other. One is field study, and I'm not going to talk too much about that. But probably most of what we're doing is dealing with patient sample and trying to understand the disease itself. But then that translates back and forth with laboratory study. So some of the parasites I'm studying cannot be grown in the laboratory, so we're using animal models. But otherwise, we're using, for example, in vitro culture for Plasmodium falciparum. And the idea and the general way my lab works is we either observe something weird in the patient that we then try to look in vitro to better understand it with better parasite, simpler model, and then feed that back and forth. So a lot of the technology development happen in the laboratory, so that's what I'm going to focus on today, with the hope that we can then translate that to field studies, better understand the disease. All right, so I'm going to start with gene expression. I'm going to talk about what most of my lab works on, which is Plasmodium vivax. And that's one parasite we cannot grow in culture. Some people here manage to do it, we can't. Most people cannot, so we have to rely on patient sample. So for the longest time, it was really difficult to do analysis of gene expression for Plasmodium in general, because you had to deal with the parasite from the patient. And if you take blood from the patient, what you will end up with is a lot of host contamination. Taking the blood, you have a lot of white blood cell on there that will completely swarm the signal. On top of that, we have another level of complexity, which is that most infections for Plasmodium vivax are polyclonal, which means that when you're sick with malaria, you're not sick because of one single parasite, but because you have multiple parasites inside you. So typically, when we sequence patients, we observe multiple genetically different parasites in the same patient, and that messes up everything. And then the parasite, when we take them from the blood of a patient, in our species, there's no sequestration, which means that all these life cycle, all these life stages are present at the same time. So we're going to have parasites that are just infected in a red blood cell, that are like trying to activate all their metabolism to start like eating hemoglobin. We have parasites that are actively dividing, and then parasites that are getting out of the red blood cell. And each of these stages have its own gene expression profile. They are very different from each other. I'm showing you a old study that they found in falciparum in vitro when they synchronize the parasite at different time points to show that, yes, the gene expression changes as you go through that life cycle. The problem is if we deal with patients, we cannot control for that. And so when we're comparing patients, we don't know what we're comparing because each patient will have a different proportion of these life stages. So trying to understand that is an absolute nightmare. And for the longest time, nobody wants to do gene expression study of Plasmodium because of that. So the first thing we did is we got really excited when the single cell technology came along. The biggest difficulty of single cell is single cell isolation. That is done for us. That is the one thing where it's actually good to work on malaria. The parasite is single cell in a red blood cell is done for us. So we use that approach to characterize the different stages and what are their gene expression profiles using single cell. It works beautifully. Again, we try that not with patients because shipping blood from where we work in Cambodia is an absolute nightmare. We get water when it reaches Baltimore. So we infected some monkeys with the human parasite, get their blood, did single cell. And you can see here, one dot is a single cell that we characterize by gene expression. And we can recapitulate the whole life cycle. So we have parasite in red that corresponds to early trophozoite that then move to Kaiser, and then we have sexual form that are in a different plane with completely different gene expression profile. So it looks beautiful. And now we actually have a good characterization of what is the gene expression profile at one stage. One thing that we really struggle at the beginning is all that data that I'm showing you on the right based on microscopy, which is what most parasitologists do. And when they characterize the parasite, they describe them as discrete entities. So you're either a ring or a schizont or a troph or a gametocyte. But what happens when we look at a gene expression is you have continuous variation. It's not different clusters that are completely separately when you jump from one to the other, is you really have that continuous development, which is what I would have expected, but it makes it very difficult to talk to people that do microscopy because I don't know what they're talking about. I don't know what are the skies on that plot. But that's one thing that we learned, that there's continuous difference. Now, the technology we use for that experiment is the 10x genomics. So I'm not going to go into too much detail, but the idea is you capture your mRNA using the poly A tail, you make cDNA, and then you fragment it. You're only sequencing the last 300 base pair of each molecule. So all the reads that you're generating with that technology should come from the last 300 base pair of each transcript. That didn't quite work in our case. So that's a snapshot of the data. So that's the read we're getting. And you can see these very nice peaks. So telling us, yes, we're getting the end of the transcripts. But they don't match with the annotation. 30% of the reads that we were getting mapped 5x gene. Most of them didn't. And some of the reads mapped really far away from the annotation. And that's a general problem that we're dealing with incompletely characterized genome. It's very easy. It's relatively easy to call coding sequences. You do enough prediction and know where the coding sequences are. Predicting the UTRs, the untranslated region, is a nightmare. So when we are getting a profile like that, we can easily see the peaks, but we don't know what gene do they correspond to. Is that there's multiple isoform, or is there some misannotated gene? So that really bothered us. So the data I showed you before, when we do the single cell analysis, we just ignore genes. We did our analysis per 500 base pair of the genome. We don't care what the genes are. We just want to look at who's expressing what. But that was kind of underwhelming. So the next step was, can we use libraries we made during the single cell and sequence them before we fragment them? When we capture the transcript, we fragment them and sequence them with Illumina, the data I showed you, but we have this full-length isoform that we synthesize. What we did is we took these guys and sequenced them with PacBio, trying to get full-length isoform from the single-cell data directly. Just to show you an example. So each read here, each blue bar is one read we're getting, and you can see that we're getting these like several KB reads that cover the entire transcript, and you can recover the nice splicing pattern that you have in the annotation. But one thing that was really apparent for us at the time, it's like the UTRs were completely misannotated. The annotation stopped at the coding sequences and totally ignored the fact that there was like a 500 base pair UTR behind it. And that explains why our single cell data map outside. Now, one thing you notice is there's also a lot of introns in the UTRs. Okay? And we see that here, we see that some here as well. We have no idea why. And that's something that we're trying to understand, is what is the role of this UTR, why sometimes are they spliced. Try to look at correlation with expression, we don't see anything. But that's an observation we have here. We also have some isoforms. So it's not one gene, one transcript, which was really the dogma for Plasmodium. But here I'm showing you an example where the protein coding gene, you can recover it very nicely, but you're actually starting transcription at different promoter at different places in different stages. But you have some isoform, and you can see that some of them are stage specific. They only express in some parasite at a certain stage of their development. We have very few examples of isoform that affect the coding sequences, but we have a ton of examples of different form of the gene, different UTRs at different stages. And again, we don't understand how it's made and how it's regulated, but if we want to understand how transcription occurs in Plasmodium, we need to take that into account. All right, so that's published. I wanted to go into something new. One thing that when we did the PacBio sequencing, that data, one thing that I haven't talked about, and it's like one sentence in the paper that we try to query, that 40% of the reads we were getting were not mapped to coding, annotated protein coding genes. Okay. A lot of them were antisense. So we really struggle with that. And I was not convinced for the longest time that what I was getting, these 40% of reads, were not an artifact coming from the reverse transcriptase. We were sequencing the other strand. So it really bothered us. So we saw that maybe a lot of non-coding RNA in the genome that were not well characterized. 2 of them have been followed up in Plasmodium, but that's all we know. So we tried to look at that, but we wanted to avoid the reverse transcriptase. And really to emphasize how difficult it is to do in Plasmodium, because the genome is so gene-rich, you always get transcription somewhere. And they always overlap with some gene. And on top of that, we have this antisense transcript that I was not convinced were real. So what we turn to is Oxford nanopore direct RNA sequencing. So all the technology we had until 5 years ago, when we sequence RNA, we actually don't sequence RNA. We sequence cDNA. So we introduced the RT. Oxford nanopore is the only one when you can actually sequence the RNA molecule directly. There's no RT. So if I see an antisense transcript, I know it's an antisense transcript. It's not an artifact. And the way it works, I'm going to go very quickly through it, is you ligate motor protein to your RNA using the poly-A tail. That motor protein guides the mRNA to a pore on a membrane. And we usually do a cDNA synthesis to disentangle the RNA to remove secondary structure, but we're only sequencing the RNA. So the motor protein guides the RNA molecule into a pore and then throw it through it. And the motor protein is really a helicase. So it just unwinds the mRNA. And as the mRNA goes through the pore, we're measuring the current. We can use that current to infer what bases is this mRNA made of. So that's an example of what the data looks like. So we're sequencing here, you have one gene going in that direction. That's all on Plasmodium falciparum because we need buckets of RNA to do that experiment. So we cannot do that on Plasmodium vivax. So we tested that on falciparum. So we're sequencing from the poly-A tail and going through it. And again, you can see we get these beautiful long reads. Some of them end earlier, and we don't know if it's because the pore is clogging and the sequencing stops, or because we're sequencing degraded mRNA. And that's one thing that we're following up in the lab. But you get these really long transcripts throughout with a bias on the 3' end. And then we had this semi-complicated pipeline to try to understand which reads are truly non-coding RNA. And really the difficulty is because we have some fragmented read. If the fragmented read comes from here, we might call that as a non-coding RNA when it's just part of a misannotated mRNA. So we went through different iterations to make the annotation of Plasmodium falciparum mRNA better, and again, extend the UTRs and then call what is not coding and use that as non-coding RNA. Just to show you a couple of examples, so that's RNA that we generate at the trophozoite stages. So you have one mRNA going in that direction, one mRNA going in that direction. They correspond to the annotation that we have in PlasmoDB. But if we look at the gametocyte, we get a lot of reads supporting something in the middle. Okay? And that one has no coding potential. So it doesn't encode for more than 100 amino acids, has no start and end. It's spliced, okay, we don't know what it is. And one thing that was curious is we see a lot of examples like that, and you can notice that when that non-coding RNA gets transcribed, we don't see the mRNA on the same strand. And here, I can make whatever story I want, but what we're wondering is if that's one way of maybe regulating gene expression. That transcription of that gene might be regulated by transcription of that gene. If you start transcribing that mRNA, that non-coding RNA, you block access to the promoter of the mRNA. So that's one example of intergenic. Again, we have like dozens of those, but the majority of them are actually antisense non-coding RNA. That's really what bothered me earlier. So I'm showing you again the trophozoite. You have one gene that is going in that direction, that is spliced, that is annotated, that corresponds to the annotation. In the gametocyte, we don't see that gene being transcribed. We have very little evidence of gene expression from that strand, but from the other strand, we have a non-coding RNA that is going through. That is also spliced, and that's why when I was getting that from PacBio, I was not tracking that data. I was worried that was an artifact of the reverse transcriptase of that gene. Here I know that molecule is being transcribed from that strand because I don't sequence cDNA. And again, when we see the antisense, we don't see the sense being transcribed. That's one of the genes that is most studied in Plasmodium falciparum. It's the AP2 gene. It's a transcription factor that regulates gametocyte development. So in trophozoite, it's expressed. It's not expressed in the gametocyte because they already differentiated. But then we see again non-coding RNA going in the other direction. And when we look at multiple stages in the time series of differentiation of gametocyte, we see that when the antisense starts being expressed, you have an anti-correlation with the expression of the sense transcript. So we're following up on that, but now we're wondering is maybe transcription is regulated by non-coding RNA, maybe more than transcription factor, or maybe they overlap to some extent. But that's a big factor that we have to take into account to understand that. So we've generated a ton of data. I'm not going to show you everything. One thing that came out of the analysis is we roughly have the same number of mRNA transcribed at different stages, but the gametocytes seem to have a lot more non-coding RNA. And that's interesting because the gametocytes are at this really weird stage when the parasite become quiescent. The gametocytes are the form that's going to be taken by the mosquito. But until they are taken by a mosquito, which can take weeks, they have to be dormant. So you have these parasites that are metabolically inactive, and at that stage, we see a lot of non-coding RNA. And we're wondering, okay, that. So the antisense RNA might be a mechanism to regulate transcription overall, but because we see a lot more in gametocyte, we're wondering if there might be also another population of non-coding RNA might be involved in mRNA stability. If you have a parasite that is dormant for several weeks, you need to inhibit translation, and you need to make the mRNA stable. Maybe these non-coding RNA are important for that. Okay, so that's why we're going now with the experiment in collaboration with Manuel Linas, when we're trying to overexpress either ectopically or using transgenic some of these non-coding RNA, if they affect the transcription of the sense RNA. But we're also doing some RNA duplex sequencing when we're isolating RNA duplexes to see what are the target of these non-coding RNA, and do they actually stabilize mRNA in the gametocytes. All right. I need to finish in 15. I ask him that.
Something like that. Yeah.
Something like that, yeah. I ask him that. All right. We're gonna have a question. I can come back if there's some extra time. So I showed you some data on what we're doing in terms of basic biology understanding of gene expression regulation. Most of what we do is actually working in Cambodia with Plasmodium vivax patients. And we recently finished a clinical trial where we recruited and followed like 160 patients that were infected with Plasmodium vivax. And as part of the project, we sequenced the entire genome of all these patients. So you have to remove the white blood cell, but then you take the blood that contains only the red blood cell and the parasite, and we sequence that at very high coverage. And one thing we always do is look for sequence rearrangement. Okay, so we have a genome, but we know that genome varies. And one thing that is really important is because the genome is haploid in the human host, if you have a deletion, it really matters. If I have a deletion on one of my genes, my other chromosome is unlikely to have that deletion. I still have a copy. If you're haploid and you have a deletion, it has a major effect. It's gone. The duplication is the same. It decreases dramatically the copy number. So we use the read pair arrangement to look for sequence rearrangement. So you can see that in the case where there's a deletion in the sample, when you map that back to the reference genome, the read pair that should be 300 base pair apart from each other might map 5 kb apart from each other because that sequence is missing. We can use that information for finding deletion, inversion, tandem duplication. Okay, so we did that. And so we did find, again, plenty of rearrangement, but we had like 36 in these 160 patients. We had only 36 common insertion and duplication. So rearrangement that involved more than a quarter of our patients. Most of them were located to multi-gene family. Okay, so we have like these families of genes that are very similar, often in tandems, and then when during replication, one gets skipped, one gets added, so on. So these are not uninteresting, but difficult to make sense of and try to understand what's happening. Out of these 36 insertions, there was a single rearrangement, a deletion of an 837 base pair. That was the only one that affected a unique gene, a gene that is present in a single copy in the genome. And that gene, the MDR1 gene, multi-drug resistance 1. So it's one of the most studied genes in Plasmodium. It's involved in Plasmodium as well as in fungi with a lot of drug. It's a gene that makes a transporter and basically pumps the drug out. So we were excited with that the only thing we found in that entire collection was right next to a gene that is involved in drug resistance. was present in 80% of the isolates. That worried me a little bit that we've been sequencing Cambodian isolate for 10 years and we never paid attention to that. But here it was present in 80%. So I'm showing you the coverage here across like one isolate that doesn't have the deletion. That one has a deletion, but some of the clones don't. You can see the reads that are like far away that we flag as indicative of deletion, but we know that there's probably like something at 10% that has the complete sequence, while that one is completely missing. And it's right after the coding sequence of the MDR1. So it doesn't affect the coding sequences. So like, is that really exciting? To check that, the next thing we did is we screened data from the Sanger Institute where they sequenced like 800 isolates from across the world. Okay. And so we went through that and we did find that their Thai, Cambodian, and Vietnam isolates also had that deletion. They didn't follow up on it because it was non-coding. Okay, didn't know what to do with it. It was also present in some South American isolates. So that really confused me. I could not quite understand how you can have a deletion that jumped from like South America to Cambodia or the other direction without anything in between. But when we actually look at the data itself, there's a deletion in the same location, but it's not the exact same one. It seems to be slightly shorter. So we had one of these isolates in the freezer, something related to one of these isolates, the reference genome from Plasmodium vivax. The early reference genome from Plasmodium vivax is that one. And that's why we missed it before, because we use a reference genome that had a deletion. When we mapped all the reads we were generated onto that reference genome, we didn't notice that they also had a deletion. In between, in the last 5 years, they switched to reference genome, or reference genome that doesn't have a deletion, and now we did it. And again, it's just a cautionary note of like, talk about next generation sequencing as like these great agnostic approaches, you can discover new things. Yes, it's true, but only if you're smart. If you don't know and you bias all your analysis with what is in there, you can only find what is present in that gene. So what we did is we designed primers surrounding the deletion, amplify that by PCR and sequence it, and indeed, here we have an 837 base pair deletion. Here we have another deletion, shorter, around 400 base pairs. So that got us excited. First, let's explain why we see a deletion in two different locations of the world. The fact that we have two independent deletions that occurred at the same region tells us that maybe it's important. We have so few deletions that rearrangement in general that we see in our parasite. The fact that we see twice hitting the same region independently tells us that it must matter. Okay? And we have RNA from all these patients. Now we're like, okay, from the patient that don't have the deletion, if we do RNA sequencing, that's what we're getting. Okay? That's the gene that is being expressed on that. When you have the deletion, it's 25 base pair past the coding sequence, but it's right in the UTR. So you end up with a different UTR transcript. We have a deletion that affects the non-coding part genome, but affects the gene expression profile. That's cool, because now we have a link to maybe that deletion is functional because it makes a different mRNA. Let's keep that part here that might have some important regulatory element, and we add a new section of it. You can see the South American isolates have a different UTR as well. So we have different isoforms that are being transcribed, and that deletion is actually associated with a higher expression of MDR1. So we just looked at the coding sequences to account for the differences in length on how much mRNA is there for that gene if you don't have the deletion. Yeah. And if you have the deletion after the gene, you end up with twice as much mRNA. Now we're really excited because we don't know what's happening, but you have twice as much mRNA, so you might potentially make twice as much protein. You might pump the drug twice as fast. The example that we have on MDR1 falciparum is a copy number variation of the entire gene that has been associated with making more pump and more drug resistance. So our assumption, and we're trying to test that now, is that when you delete that section, whether in Cambodia or in South America, you affect the regulation of that mRNA. and you actually make it more stable, potentially. That's our hypothesis. Because it's more stable, you get more mRNA over time. Maybe that makes more protein, maybe that makes more form. What we did, so we look ex vivo, which is isolate, try to see how do they respond to mefloquine, which is one of the anti-malaria drugs. And you can see that when you have the deletion, the parasites are much less susceptible to the drug. We're missing one part. We're really trying hard to make that mechanistic link between more mRNA and more susceptibility, but it's an almost complete story. And when we went back to the freezer and look at parasites that have been collecting for the last 15 years, you can see that the proportion of parasites that carry the deletion increases over time. Okay. And that corresponds to when they started using mefloquine in Cambodia in the field. We think that what happened is they switched the drug regimen. They introduced mefloquine. That deletion that was already present before now got selected for. Increase in frequency because it gives them an advantage. And it's a multi-drug resistant cap. It's just a pump. So it's a very non-specific mechanism of getting resistance to anti-malarial drug. And again, now we really try to have to understand what is happening. It could be that it affects directly transcription. That's not our primary hypothesis because it's in 3' end. So it would be a bit hard for us to explain how it affects transcription, but maybe it's affecting mRNA stability and decay or translation. And again, just emphasizing that we need to look outside of just the coding sequences because a lot of things that are important are outside. And again, this deletion was present in the Sanger data published like 7 years ago. Nobody followed up on that because it was non-coding and couldn't link that immediately to something. But summarize, again, it's not surprising if you're coming from a human or mouse or model organism background, but or unicellular or eukaryote, that's people are still struggling with that one gene is not one transcript, one protein. There's lots of different isoforms that exist that are being used at different times. We think most of that has to do with 3' and 5' UTR that we vastly ignore, and we really need to take that into account, that there is a reason why different stages use different 3' and 5' UTR. Show you an example with the MDR1 story that these matter. These might have some functional consequences and some clinical relevance. And now we're really trying to follow up on that and like reconciliate the UTRs with the non-coding RNA. I think there might be a direct link that there's a lot more non-coding RNA that we thought, or at least that we believed. They might affect transcription. I think the antisense transcripts are a really good candidate to look at that. So we're overexpressing some non-coding RNA to try to see if it affects the mRNA on the other strand. But they might also affect protein stability and translation. So I'm gonna stop here, acknowledge the people that have done the work. So all the single cell was done by Brittany that just moved to the University of Chicago. Yana is responsible for all the ONT sequencing, and we've been really playing a lot with that technology. I think there's a lot of things that we can gain from that technology. We got diverted with looking at ribosomal RNA and non-coding RNA, while our initial premise was to look at RNA modification. So we're going to have to come back at that at some point and try to see if this matters for gene expression regulation. And Katie is a graduate student that is looking at our Cambodian sample and trying to understand if we can go the other way and start from what's happening in the patient to try to understand the biology. I'm happy to take any question.
We see common variations among the isolates. Do you believe those variations are occurring or emerging independently, or it's linked to a parasitic lineage that?
So the other sequence rearrangement that we're going after. So these ones are tricky. A lot of them are this very large multi-gene family where you have multiple similar copies of the same gene in tandem repeat. Everybody in the field has been staying away from that gene family because we can't, with the short read technology, tease apart if it's a real rearrangement or if there is a mismapping artifact. Now that we have long reads, we hope we can get to that. I have one of my postdocs that is really excited about that, and she's somehow convincing me that it's worth taking a shot at that. There's like 20% of the gene in Plasmodium vivax belong to one of these gene families. So they must do something. There's so many of them that they must have it's a pain to study because each genome is different because there's a lot of non-homologous recombination that happen. So if you want to understand that gene family, you have to sequence the isolate that you're actually working with. And then you need a long read to know which one is being transcribed. And because we now have the technology to do that, it's very feasible to de novo assembly from field isolate. And we can do long read sequencing. We're going to start looking at that. But right now, I don't know if they even know. Yep.
Okay, hi, thanks so much for this talk. I was just wondering, have there been any studies that sort of look at differences in like chromatin between these different stages of the parasite, and if that would give you any insights into how the regulation is working?
Yeah. So yes, it is really complicated. So we've never been able to do ATAC-seq or DNase one experiment, ever. It drove me, we wasted so much time and money on that. There's one group that claimed they can do it, not entirely convinced. Part of it is the fact that you have multiple stages, and some of them, within 24 hours, they go through a DNA replication. If you don't have a really nicely synchronized culture, you're going to have mixture of chromatin that is closed and chromatin that is open. I'm also not convinced. I suspect that the histones don't bind as strongly in Plasmodium as they do in human. When we do ATAC-seq, we get a beautiful profile from the white blood cell that we didn't remove, and nothing from the Plasmodium. So it was not just a technical artifact. So trying to do that, maybe ChIP-seq works slightly better, but you need to know what you're looking for. So again, that's one thing that I'd like to look at. Maybe what didn't mention is like when we do the duplex sequencing for trying to find DNA, RNA, one thing we want to do in parallel is look for whether the non-coding RNA might bind directly to the DNA sequence. We're going to look for heterologous like DNA, RNA duplexes. That might get us to there. It's tricky. Hi-seq works really well, but it doesn't have the resolution we need for that. So it's one thing that has been understudied, I would say.
Okay, yeah, thank you.
Yep.
I just wonder, have you identified any transposable element in these species?
No, there's not. As far as I know, in Plasmodium, there's no. Is that correct? I think it's correct.
Another question is, I saw that you mentioned two Plasmodium. One is really high GC rich, another is not that GC rich.
Yeah.
Do you have any explanation?
No. It's just separated for so long. They're really different. There's some people that I've looked at the evolution of this genome that could answer that better. The coding regions, I mean, they cannot be that AT rich. You can't make a protein coding gene with just A and Ts. But as soon as you get out of the protein coding region, basically A's and T's all the time, which makes it really difficult to try to understand how you regulate transcription. Because it's really hard to find like a binding site if you only have A and T's everywhere. So the one I'm mostly studying, Plasmodium vivax, has a more friendly GC, so it's easier, and we hope that there's enough diversity in the base composition that we can identify some motif, but we really don't know. Some of the Plasmodium genomes have like absolutely insane AT content, but some are normal and they behave the same way.
Okay. The last question is, have you identified the centromere in this small species, or did you look at when the genome sequence? Do you see some region with really low gene density?
The telomere and the centromeres, I don't know if they're really well sequenced. We're trying to people here might know that better because they did some recent genome assembly. We're just doing that, so I haven't looked at the sequence. Wayne, do you want to say something about that? You might know the answer better than I do.
Yeah, in my lab, we did a lot like a centromere assembly and some things. But theoretically, because the genome is really small, so if you have long reads, theoretically.
I know. So let me answer something else. So one thing I didn't show you is one of my postdocs works on Trypanosoma brucei. And when she came to my lab, she's like, "Oh, we don't have a full genome." I was like, "I can do it, easy. We can sequence anything." It turns out we cannot sequence anything. So it's really easy to assemble like what they call the core genome, which is where all the unique genes are. As soon as you get into this weird region that are subtelomeric or centromeric or past, like even like mini chromosome outside, it's an absolute nightmare. And it's not like, I thought it was an assembly issue, that we were not able to put them together. It turns out some of these regions are just hard to sequence. And at least in T. brucei, some of the bases on the DNA are heavily modified. And we think that that modification blocks the sequencing. That we see like the sequences from nanopore, they get stuck because it's too big. So there's some complication here. For a lot of these organisms, not Plasmodium, but for a lot of these similar organisms, we don't have a good genome yet because of that. It's actually challenging.
So for the centromere question, the import connections care about them, so it's not really doubtful. But if you actually look at that, there's a specific region. It's most likely the centromere. It's going to obviously follow up, but you can definitely see it.
There's a massive bias in the community into what matters before. So it's really hard to convince people to look outside of protein coding gene or coding sequences. So the telomere and centromere is way down the list. So hopefully gonna get there, but it's gonna take a, yeah.
So Dr. Jesse Kissinger is asking a question online. To what extent do you believe that genome challenges are caused by sequencing populations?
So that again, that
To what extent do you believe that genome challenges are caused by sequencing populations?
that we have a bias in what we sequence? Is that the question?
I think in terms of like clonal parasites versus.
Heterogeneous.
Yeah.
Okay. So, so if that's the question, I don't know, maybe Jesse can clarify that. But the problem is when we work with field isolates, I told you, we rarely have a single parasite in there. The long reads might be an issue. So it's easy to characterize what polymorphisms are present in a different isolate within one sample because we sequence at very high coverage, sequence at 100, 300x. So if something is present in 3%, it's going to be represented in these reads. The issue we have is reconstructing this haplotype. If you have 3 genomes together, knowing which A here goes with which T here is an absolute nightmare. So we tried different approach to solve that. If there's a lot of SNPs in the same region, we can walk along the molecules and reconstruct haplotype. But at any point when you have places when there's no diversity, we lose that. The long read sequencing might be a way to get that, to get like phasing information directly. It could be a major issue if, that's maybe what Jesse is after, is some parasites or some genotypes predominate, grow faster. There will always be more abundant. Then we would have a bias in what we characterize. That the lowly abundant parasites, they're gonna be swamped and we're not gonna capture that. It's true to some extent, maybe not. So within a species, I don't know. If I told you that when you're infected, you often have multiple parasites. Often we're infected by different species of the parasite. And so Vivax and falciparum are the main one, but there's something like ovale and malaria. Often we detect them after drug treatment. Someone comes with falciparum, we treat them, and then we see some parasite left that is actually another species that is less susceptible. So that might create some biases in that. Yeah, I'll try that as
She was asking about sequencing like populations instead of single cells too. So I think you addressed the correct thing.
Yeah, I mean the single cell. We actually So single cell is really cool because everybody wants to do single cell, toy that we have. It actually gives you really an impressive gene expression profile. You get very shitty data compared to a bulk RNA-seq. The advantage is you get a single cell. Most of the time you don't, but we're actually using the single cell RNA-seq technology more to separate the parasite and genotype them. I didn't show you that, but we have like a paper last year where when you sequence RNA, single cell data, still sequence based. So we're sequencing T parasites. If there's a polymorphism at that locus, you can genotype each individual parasite. We're able to use the single cell RNA-seq data from, we've done that in monkey and we're trying to do that in patient, but to separate the different genotype and say that cell here is clone A, and that cell is clone B, because we have the genotype information. So that might be one way where we're using the technology for a different purpose, but I think that's more interesting because we can disentangle that complexity. Yeah.
Yeah. What about single cell sequencing? Is there single cell sequencing of the host cell or parasites and single cell parasites?
So everything I showed you is single cell of the parasite because we sequencing the host and the parasite together. But the host in this experiment is a red blood cell. Nothing exciting in there. So we looked at that. That's something that really confused us initially. Plasmodium vivax, which is the data, supposed to infect young reticulocytes that still have mRNA and the nucleus gets expelled. We never find RNA from the host. What we think is going on, and there's one paper in the literature saying that when Plasmodium vivax infects a red blood cell, it actively pumps out everything that is in there. That's probably why we are not seeing the host in that case. In the liver or other stages, that would be very interesting. That's what I want to do, is look at the host and the parasite, how do they affect each other? But here it's just a boring red blood cell that doesn't do anything. So we could technically do that, but not with that data. Yep. Go ahead.
You showed that there were genes where the antisense RNA was expressed and the mRNA was not expressed.
Yeah.
In defined stages, those were defined sets of genes.
Yeah.
Was there any enrichment of certain Go terms or anything?
It's really, so we haven't done it, and the reason we haven't done it is the annotation is so bad that you never get I never get something meaningful from the annotation. So half of the genes in the plasmodium, whether it's falciparum or vivax, their annotation is hypothetical protein, conserved protein. We have no information. So the geoanalysis is not very interesting, and on top of that, it's the annotation is heavily biased. So the geo for the genes we have, they're all extremely conserved genes for glycolysis. They conserve the cross and you can transfer that annotation. Or some specific pathways that people have been studying for 40 years, like red blood cell invasion. So you start with a biased data set. So I'm reluctant to push that through because I don't think what I'm getting is meaningful. That's probably the main challenge of Plasmodium genomics right now, that we don't know what these genes do. When I find a really cool gene, so I get excited when it's MDR1. It's like, yes, I can actually follow up. I have a story there. If it's hypothetical protein 3, like out. So and that's half of the gene.
So it's 12:25 already. We have to wrap it up here. So let's give the speaker another round of applause.
This is a lot. Okay.